Everyone, um, am I audible and visible? So, yes. Good afternoon. Welcome to this FMG recall session. This session is to discuss biochemistry MCQs that were asked in the latest FMG July 2025 exam. Before we start the session, let me tell you all, you have done your part, right? The pen is down, the exam is over. So it is now time for you to take a deep breath, right? And to just reflect and not to regret, right? The purpose of the session is only to reflect on how the question paper was, what kind of questions were asked, how was your performance, so that you clearly understand where you stand, so that you can plan your future, right? It is definitely not to regret. So have an open mind during the session. Just try to understand how many questions were asked, what kind of questions were asked, what are the probable answers for all these questions, what did you answer for those particular MCQs, so what is the approximate mark that you would get. That's all. Just analyze. It's not to regret, right? So yeah, good afternoon. Biochemistry was easy, right? Yes. So many of the students had given me this feedback. So based on the poll that I conducted on my Telegram group and based on my interaction with few of the FMG aspirants who came and spoke to me after the exam, this is what I found. The paper was moderate and biochemistry was easy, though there were totally 19 MCQs asked in biochemistry and clinical biochemistry. 15 of those MCQs were case-based MCQs, right? It's interesting to know. And then 2 MCQs were image-based MCQs. Actually, only 2 MCQs were fact-based, right? So this should clearly indicate how your preparation should be for your future. Well, not for those who have written the exam, those who are future FMG aspirants, you have to learn all subjects conceptually, right? And then 6 MCQs were based on inborn errors of metabolism. And almost all MCQs were based on previous year topics, right? I won't call those as previous year MCQs. All these MCQs are covered previously as topics, okay? So this is what my observation or my analysis, overall analysis of the paper. Okay, so now let's start discussing all these 19 MCQs one by one. I will not take much of your time. I will only tell you the right answer and then I will exclude all wrong choices. Okay. So this is the first MCQ that we are going to discuss today. This is based on genetics. Yes, this we have discussed multiple times. A person presenting with multiple colonic polyps has been diagnosed with colorectal cancer. There is a strong history of hereditary non-polyposis colorectal cancer. And what is the DNA repair mechanism that is defective in this condition, right? That is the question. So if it is HNPCC, right? How did I ask you to remember this? Double C is double M. What does double M stand for? Mismatch repair defect. So HNPCC is caused by a defect of mismatch repair. So answer for this question is very good, Pookie Arshad. Very good, Deepinder. Yes, the answer is mismatch repair. Okay, let's go to the next question. And this we know, nucleotide excision repair. There is an X that causes which disorder? Xeroderma pigmentosa. Okay, so let's go to the next question. A man presents with high VLDL and chylomicrons. Okay, both VLDL and chylomicrons are elevated, meaning triglycerides are elevated with eruptive xanthomas. I've told you multiple times, hypertriglyceridemia will be indicated by the presence of eruptive xanthomas. What is the most likely diagnosis? What is your answer, everybody? The answer is familial hypertriglyceridemia. I've told you in familial hypertriglyceridemia, both chylomicron and VLDL levels are high. Both carry triglycerides. So serum triglyceride level will be elevated. Whenever triglyceride levels are elevated, you will expect eruptive xanthomas. So what is the right answer here? The right answer here is choice C, familial hypertriglyceridemia. Okay, and I've also told you that this is an autosomal dominant condition, right? So why did I exclude familial hypercholesterolemia? If it was familial hypercholesterolemia, the classical feature that they would have given is elevated LDL, and they would have told you elevated cholesterol level, right? And you will not see eruptive xanthomas, you would have seen tendon xanthomas. That is why we have excluded what? Choice D. Right? Eruptive xanthomas are different from tendon xanthomas. Whenever it is eruptive xanthoma, it is hypertriglyceridemia. Do you understand that, Saf? And then why did I exclude choice B? The choice B can be excluded because familial dysbeta lipoproteinemia would have presented with broad beta band, right? Broad beta band on lipoprotein electrophoresis, and remnants level should have been elevated. There is no expectation for elevation of VLDL and chylomicron in this condition. That is why familial dysbeta lipoproteinemia is excluded. Okay. LPL deficiency was there. Okay. Had they given you that there was LPL deficiency, and if the choice had familial chylomicronemia syndrome, then that is the right answer. Do you all understand that? Had they given LPL activity was low, lipoprotein lipase activity was low, and if the choice had familial chylomicronemia syndrome, that will be the right answer. Okay. Yes. Now the next question, a breastfed infant. The first clue that you're given is a breastfed infant presents with lethargy, hepatomegaly and cataracts. Which of the following enzyme deficiencies is most likely responsible for this presentation? What will be your answer? The answer is galput deficiency because the diagnosis is classical galactosemia. How did I exclude other choices? This is not galactokinase defect. Had it been galactokinase defect, that should have been only cataracts, right? That should not have been hepatomegaly. Because they have given you hepatomegaly, I am excluding choice A. Okay. Why is it not choice C and choice D? Both choice C and choice D are related to fructose metabolism disorders. Had it been fructose metabolism disorders, the history would have told you that the child was perfectly normal until the child was mother fed. Only after transition from mother's milk to infant feeds, all these started appearing. Had that been the case, I would have chosen fructose metabolism disorders. Here, the history clearly states the child is mother fed. So, mother-fed child presenting with cataract with hepatomegaly is classical galactosemia caused by galput deficiency. You didn't have this question? Oh, okay. But I was given this question by many students. Okay, if it's not there, let's exclude it. Which vitamin deficiency can lead to? Okay, we'll exclude that question. Okay. So, which vitamin deficiency can lead to lactic acidosis? Haven't we discussed this? Was this question asked? Yeah, what is the right answer? Thiamine. Right? Because when thiamine is deficient, which enzyme is inactive? Pyruvate dehydrogenase complex is inactive. What is the function of pyruvate dehydrogenase complex? It should convert pyruvate to acetyl-CoA. So whenever there is a deficiency of thiamine, PDH complex is inactive. Pyruvate is not converted to acetyl-CoA. So no other go pyruvate gets converted to lactate. So in lactic acidosis, what should be supplemented? Thiamine has to be supplemented so that pyruvate gets converted to acetyl-CoA. It's not converted to lactate. Okay. So answer is choice A. Very good. All of you are right. The next one. So this we have discussed so many times, right? Even in the LRR session, we discussed about blood collection tubes, right? In the image-based session, I told you, I clearly showed you all images. For coagulation studies, I told you C for C, it is citrated tubes. Citrated tubes are in which color? Blue color. So what is the right answer? The right answer is choice A, right? And what did we learn about green? Green has heparin. What is heparin used for? Heparin is used for quicker biochemical analysis, or it is for molecular diagnostics, or it is for cytogenetics. Okay, that's for heparin. Lavender. All of you answer about lavender. Lavender is EDTA tube. What is EDTA tube used for? Complete blood count. Pathology, right? Complete blood count. HbA1c. Remember, or parathormone, ACTH, or ammonia. What is red used for? Red is silica, right? I gave you a mnemonic, red silica serum. This is used in biochemistry. In biochemistry for what? RFT, LFT, hormone assays. Good, excellent. This question was there, right? A patient presents with fatigue, anemia, and hepatomegaly. Hemoglobin electrophoresis reveals the presence of HBS, right? Which means the person is suffering from sickle cell anemia. What will be the appropriate treatment? Obviously, the answer is hydroxyurea. Right, the answer is choice A, hydroxyurea. What is your aim? Your aim is to increase fetal hemoglobin. When you increase fetal hemoglobin, sickling can be avoided. Right? Will you ask for blood transfusion? Yes, you can ask for blood transfusion, but in acute phases. Right? Will you ask for iron supplementation? So blood transfusion is not for long-term treatment. For acute sickling crisis, probably blood transfusion can be helpful. But iron supplementation is contraindicated. That will add iron overload. So this is contraindicated. Vitamin B12 supplementation has got nothing to do with sickle cell anemia. Okay, yes. So this question was asked, right? I'm going to ask this question for every question. I hope this was asked. A 17-year-old tall male presents for a routine checkup. He has long limbs, arachnodactyly, hypermobile joints, hyperarched palate, and high arm span. Right? So these two are the clues. High arm span, arachnodactyly. What is your diagnosis? Marfan syndrome. Right? Marfan syndrome is caused by the defect of which protein? It's caused by the defect of fibrillin protein. Fibrillin gene mutation causes Marfan syndrome. Yeah, the same question was asked twice. Similar question was asked twice. So the answer is fibrillin gene mutation causes Marfan syndrome. Okay. What is elastin gene mutation? Elastin gene mutation causes Williams syndrome. That's not Marfan syndrome. That is Williams syndrome. That is characterized by supravalvular aortic stenosis. Remember, Williams syndrome is supravalvular aortic stenosis, elfin-like faces, hypercalcemia, and developmental defects. They don't present with arachnodactyly. They don't present with long limbs. Okay. And then collagen mutation. We know collagen 1 mutation causes what? Yeah, fragile bones. What is it? Osteogenesis imperfecta. Right, collagen 1 mutation causes osteogenesis imperfecta, which is characterized by fragile bones, blue sclera. Remember, so that is not the case here. Yes. And then collagen 3 and collagen 5 mutation would cause what? Ehlers-Danlos syndrome. Okay. So the answer. So was this question also asked in a different way? A 17-year-old tall male presents with long limbs, arachnodactyly, hypermobile joints, fibrillin 1 gene was found to be defective. What is the associated disease? No, this was not asked. This is also Marfan syndrome. 2 questions on Marfan's. Good. So the answer is choice B. Similar to this, right? Yeah. This is also Marfan syndrome. Which of the following acts as a major intracellular antioxidant and helps in detoxifying reactive oxygen species? All of you tell me what is answer. The moment they say intracellular antioxidant, major, right? I would go for what? Glutathione. Answer is glutathione. Why did I exclude other choices? Yes, catalase is also a major intracellular antioxidant, but catalase is restricted to few suborganelles like peroxisome. I won't call it as major. I will call it as intracellular antioxidant, but it is restricted to few suborganelles like peroxisomes. It's not a major antioxidant. Superoxide dismutase and peroxidase. I would exclude straight away peroxidase. Because many a times peroxidase is not an antioxidant. It induces oxidative stress, right? Myeloperoxidase actually induces oxidative stress. And if you look at superoxide dismutase, it can only detoxify superoxide radicals. It cannot detoxify anything else. Whereas glutathione is a universal major intracellular antioxidant. Okay. Answer is choice A. 245 marks. Good. Options were different, but glutathione is the right answer. Okay. If you can recollect choices, I will be happy to help you exclude those choices. Which of the following genetic mutations is most commonly associated with familial cases of amyotrophic lateral sclerosis? What is the right answer? Amyotrophic lateral sclerosis caused by the defect of superoxide dismutase 1 gene mutation. Right? Answer is this. And it's the gain of function mutation. Superoxide dismutase gene gain of function mutation is the most common cause of amyotrophic lateral sclerosis. Paper was moderate, but you still did it. Very good, Ankit. CFTR gene mutation causes cystic fibrosis, right? FMR1 gene mutation causes Fragile X syndrome. And this is Huntington's chorea. Okay? Yes. A 7-year-old child is brought to the clinic with complaints of fatigue and poor concentration. The mother reports that the child has been eating non-food items such as chalk and soil for the past few months, which is pica. Right? So pica, fatigue, poor concentration, peripheral blood smear showing microcytic hypochromic red blood cells. So what is your diagnosis? Classically, it is iron deficiency anemia. Okay, had they shown you basophilic stippling? Was it given? Basophilic stippling. And had they given you that the child is taking lead chips or paint chips, then the answer is lead poisoning. Do you understand this? If the same question was asked and had they told you that there is basophilic stippling and had they told you that the child is having paint chips, then I would like to change the answer. The answer would be lead poisoning. Exam was relatively easier, right? Yes, that's what even I feel. If it's paint chips, it is lead poisoning. Okay, because I was given another question for the same history. It's lead poisoning, students. Okay. See, this is another question that I was given. A 5-year-old child is brought to the clinic with abdominal pain, irritability and developmental delay, and then bluish line and pallor, right? Bluish line is Burton's line. What is Burton's line? What does it signify? That signifies lead poisoning, because in lead poisoning, the lead in the blood reacts with hydrogen sulfide, right? Lead in the blood reacts with hydrogen sulfide that is generated by bacteria that is present in the oral flora. And that forms lead sulfide, and that lead sulfide is blue in color. So whenever the history says bluish line on the gums with basophilic stippling, then the answer is lead poisoning. In a village of Assam, a man is observed walking with the help of a stick and has genu valgum. Many people in the same village present with similar symptoms, right? So walking with the stick, Assam, genu valgum. All these mean it is skeletal fluorosis, right? What is the right answer? The right answer is skeletal fluorosis, which happens whenever the lead content in water is more than 2 parts per million, right? So this is skeletal fluorosis. It is not vitamin C deficiency, right? In vitamin C deficiency, you don't see this classical Assam with the help of stick, genu valgum. So all these point towards skeletal fluorosis. A 15-year-old boy presents with tremors, difficulty in speech and behavioral changes. On examination, he has hepatomegaly. So we know the diagnosis, right? What is the diagnosis? The diagnosis is Wilson's disease. For Wilson's disease, we discussed this in the LRR session. The definitive diagnosis for Wilson's disease is liver biopsy for copper estimation, right? So what is the right answer? It is liver biopsy. Yes, all these can be used. Serum ceruloplasmin level will be low, right? Serum copper level will be low. Copper excretion urine will be high. But the gold standard would be liver biopsy for copper estimation. Good, all of you. A family consumes a diet predominantly of rice and pulses, develops signs of muscle weakness and movement, right? So rice could be polished rice. Pulses are not rich sources of thiamine. That is when they present with dry beriberi, wherein neuronal system is affected and that causes muscle weakness. So what should be given for prophylaxis? Thiamine has to be given for prophylaxis. Okay. So parboiled rice can act as a rich source of thiamine, but if it's polished rice and just pulses, then that can cause thiamine deficiency. Answer is choice B. Very good. A 6 months old infant, this question was asked. Yeah, 6 months old infant is brought to the emergency department with lethargy, vomiting and poor feeding. The episode occurred after, all of you tell me yes or no, the episode occurred after prolonged fasting. Laboratory results revealed hypoglycemia. So what you see is fasting hypoglycemia and low ketone levels. This was not asked. Okay. If it was not asked, let's skip. But in future, if this question was asked with fasting hypoglycemia and hypoketosis, then it is fatty acid oxidation defect. Right? I told you this clearly. Fasting hypoglycemia usually is accompanied by ketosis. But whenever they give you fasting hypoglycemia with low ketone levels, because some of you are saying that this question was asked, some of you are saying this question was not asked. But the classical feature that you should observe is fasting hypoglycemia with low ketosis. It is fatty acid oxidation defect. So here the answer is medium chain acyl-CoA dehydrogenase deficiency. Okay. The answer is medium chain acyl-CoA dehydrogenase deficiency. This was asked. A 2-month-old infant presents with poor feeding, vomiting and lethargy. Laboratory studies show hyperammonemia and 3-year-old convulsion with hypoglycemia and, one second, and hepatosplenomegaly. Was it asked? Okay. Any other history apart from hypoglycemia, convulsions and hepatosplenomegaly? Did they mention anything about ketosis, all of you? Did they mention anything about ketosis in that question? If that was ketosis, it is von Gierke's disease. If they mention that there is no ketosis, then it is medium chain acyl-CoA dehydrogenase. Okay, so good. So let's finalize this question. The question history says, thanks Radhika Sharma. So the question is 3-year-old presenting with convulsions with hypoglycemia, hepatosplenomegaly, and there is no ketosis, right? So if this is the question, then it is medium chain acyl-CoA dehydrogenase deficiency, because when there is no fatty acid oxidation, gluconeogenesis cannot happen. If gluconeogenesis does not happen, that presents as fasting hypoglycemia. When there is no fatty acid oxidation, acetyl-CoA cannot be formed, without which ketone bodies cannot be synthesized. So hypoketotic hypoglycemia is medium chain acyl-CoA dehydrogenase defect. And this question, I think I got this question from many students. A 2-month-old infant presents with poor feeding, vomiting and lethargy. Lab studies show hyperammonemia and elevated levels of orotic acid in the urine. The answer is ornithine transcarbamylase deficiency. Right? What is answer? It is ornithine transcarbamylase deficiency, which is type 2 hyperammonemia. Only in type 2 hyperammonemia, not in type 1 hyperammonemia. You will see in type 1 hyperammonemia, ammonia levels will be high. In type 2, ammonia level will be high. Additionally, orotic acid level will also be high. What about choice C, orotridylate decarboxylase deficiency, that is primary orotic aciduria. In this condition, yes, orotic acid level will be high, but ammonia levels should be normal. Just because they mentioned that there is hyperammonemia and elevation of orotic acid, what is the right answer? The right answer is choice B, orotridylate decarboxylase deficiency. So Shuvankar is asking me about orotic aciduria. Just listen to this. Orotic aciduria is of two types, primary, secondary. Okay, primary is of two types, type 1, type 2. In type 1, there is a defect of two enzymes. Okay, one is orotridylate phosphoribosyl transferase defect. The other one is orotridylate decarboxylase defect. When both these enzymes are defective, it is type 1 primary orotic aciduria. In type 2, only decarboxylase is defective. Both in type 1 and type 2, what will you see? It's a pyrimidine synthesis defect. So in pyrimidine synthesis defect, only orotic acid levels are high. There is no reason for ammonia levels to be elevated. But because pyrimidine is not getting synthesized, you will see macrocytic anemia. So primary orotic aciduria presents with an elevation of orotic acid levels, normal ammonia levels with macrocytic anemia that does not respond to B12 supplementation. Okay. Now, what is secondary orotic aciduria? Secondary meaning there is some other pathway that is defective that is causing orotic acid levels to be elevated. So, secondary orotic aciduria, the most common cause is type 2 hyperammonemia, wherein ornithine transcarbamylase is defective, because of which carbamyl phosphate accumulates. That carbamyl phosphate enters into pyrimidine synthesis, wherein orotic acid accumulates. Okay, so what you see in secondary orotic aciduria? In secondary orotic aciduria, not only orotic acid levels are high, ammonia levels are also elevated. Okay, and this condition does not present with macrocytic anemia, but it will present with neurological manifestations as shown here. Right? Poor feeding, vomiting, lethargy. This means there is neurological manifestation, which means it's urea cycle defect, which means it is ornithine transcarbamylase deficiency. Do you all understand that, Shuvankar and others? So the answer is B. The clue is to check ammonia level. If ammonia level is high, it is urea cycle disorder. If ammonia level is normal, but if orotic acid level is high, then it is pyrimidine synthesis defect. A child presents with abdominal distension and liver enlarged by 8 centimeter, right? 8 centimeter liver enlargement is massive hepatomegaly. The liver is smooth on palpation. Which of the following is the most likely diagnosis? I would say it is glycogen storage disorder. Okay, what is the right answer? It is glycogen storage disorder. How did I exclude other choices? Right? So I would first exclude hepatocellular carcinoma for two reasons. One is it's a child. So the probability of hepatocellular carcinoma is low. And even in a child, if there is hepatocellular carcinoma, the liver will be nodular. If it is hepatocellular carcinoma, the liver will not be smooth. The liver would be nodular. That is why I excluded hepatocellular carcinoma. Why did I exclude lysosomal storage disorder? Had it been lysosomal storage disorder, many a times it would be hepatosplenomegaly. They have not mentioned anything about spleen here, right? That is why I have excluded lysosomal storage disease. Why did I exclude autoimmune hepatitis? Yes, autoimmune hepatitis is possible in a child, but in that case, hepatomegaly will not be massive, right? Only in extreme cases, it can be probably 2 to 3 centimeter below the costal margin, right? It's not going to be a massive hepatomegaly. That is why what is the right answer? The right answer is choice A, glycogen storage disease. So this is the last question. Which of the following substances at the given concentration makes the water unsuitable for human consumption according to safety standards? What is the right answer? Quickly you can give the answer, right? Cadmium. What is cadmium? It's a heavy metal. We don't want heavy metal poisoning to happen to people who are consuming this water, right? And cadmium concentration has to be less than 0.003 milligram per liter for it to be safe for consumption. Otherwise, it is going to cause kidney failure, right? So the answer is cadmium. But what about fluoride? We know fluoride can be till 1.2 milligram per fluoride. What was the value? Chloride can be till 250 milligram per liter. No cadmium. So what was the other choices? What were the other choices? Calcium can be till 75. Any other heavy metal was given in the choice? Fluoride, yes, but what was the value? Fluoride was abnormal, is it? Okay. I will just cross check these values, but I was given cadmium as one of the choices. That is why I chose it. But you know normal values, so based on that you will be able to choose it. Okay. Yeah. So if it's more than 1.2, then it will cause dental fluorosis. So if the value is more than 1.2, then it is fluoride. Okay. So that's about the 19 questions. I thought 19, but you said one question was not asked. So totally 18 MCQs in biochemistry, and all 18 MCQs were asked previously as topics. And so the summary is, going forward, anybody who is going to appear for the next FMG exam, do prepare all previous year questions as previous year topics, learn all topics conceptually, because most of the MCQs that were asked this time was case based. Still, those case based MCQs had classical clues which will help you to pick up the right diagnosis. They did not twist and turn it. Most of the MCQs were straightforward, so the paper was moderate. And I'm sure many of you will be able to clear FMG exam. So rest assured, be happy, be relaxed. You have done your part, and the rest is, up to the superpower who's going to help you to cross this stage and go to next level. Yeah, my best wishes to all of you. There was question on orotic acid. I answered that question out of charting. Orotic acid elevation, the answer is type 2 hyperammonemia, ornithine transcarbamylase defect. Okay. So thank you. Have a good day.